Dear students, welcome to you. In our lesson today, we start with the chapter 3 again, and we want to continue with the types of asexual reproduction in the living organism. Let's go down in our lesson today. Regeneration. Well, it is the third type of asexual reproduction. In our lesson, we need to know what does it mean and some examples on regeneration. Well, it is a kind of asexual reproduction that happens by mitosis or mitotically to the nucleus of the living organism, in which what happens, a part of body, if you cut a part of the body, okay, it grows to form a new organism, such as what? Such as starfish, like some types of worms like planarium. Let's to say it. Well, it's most common in many plants and some animals. If you cut apart a piece of the plant and you put it in the ground or in the soil, it will grow to give you a new individual. This is a simple meaning of regeneration. So it is a common in plants and some animals. For example, hydra, starfish, and planaria. Well, planaria, it's a type of worms which live in what? Live in fresh water. I mean, fresh water is mean like water opposite side of the salt water. Okay, we have three different types of water: fresh water, and type of the flat worms. We have different types of worms. We have a rounded worm. We have a flat worms, a rounded worm, and flat worms, and also we have different types of worms. Also, for example, starfish. If you cut one arm of the starfish, okay, with the central part of the central disc of the starfish, near the starfish, it has 5 arms. Okay, if you're going to cut one arm of this fish with part of the central disc, and they are separated from the mother soul, from the individual, what's gonna be happen? Well, 2 points happen here. The part which is cut, it will regenerated or compensated, and the part which is cut, it will reproduce to give us what? A new individual. So if you cut one arm with part of the central disc, the central part of the starfish, to form a new individual, what happened? The old sea star regenerate what? The cut part. And also in planarium, as we said, it's a type of freshwater worms. It gives what? A new individual. If you cut it into a transverse or longitudinal parts, both of them gonna be happen. Okay, like this picture. This is a regeneration in planarium, and actually it's a very simple picture for planarium. We're going to cut it longitudinally or transverse. Each part, it grows to give us what? A new individual. This is the meaning of regeneration or asexual reproduction by regeneration. Okay, but to compensate the missed part, this is what? This is a generation only without asexual reproduction. The capacity of regeneration. Well, regeneration, it is ability to decrease with the development. What does it mean? Let me to show by examples. Number 1, in sea star or starfish, hydra, and the planarium, they are primitive living organisms. So primitive here, it means they haven't a sophisticated or complicated systems like the higher living organism. Okay, so by regeneration, they produce a new individual. Okay, if you cut a part of it, it gives you a new individual. But in some higher living organism, but not more higher, okay, just higher than hydra or planaria, what happened for them? Well, in crustacean and amphibians, the regeneration is restricted to form what? The cut parts only. Well, the crustacean, like what? Crustacean like shrimp, like crab, they are live in water, like insects, any living organism that it doesn't have endoskeleton, but they have what? Exoskeleton. Okay, they are covered by chitinous or by skeleton, but it's a hard skeleton. Okay, like shrimp and the crab and so on. Okay, and some type of amphibians. Okay, like toad and frogs. The regeneration is restricted by what? To compensate just the missed part only. Okay, if you cut a leg or part of the toad or frog, it will compensate it. Okay, just and the missed part, it doesn't give you new individual. Okay, this is the meaning of capacity of regeneration. So in primitive or lower, it gives you what it produced to give us new individual. But in case of more higher than living organism, it just restricted to form to compensate the missed part only. While in case of high vertebrates like us, okay, the regeneration is restricted only to heal wounds, for healing wounds in the blood vessels and muscles. For example, if you cut yourself or you harm yourself in your hand, what gonna be happening for the wound? It just gonna be healed. Okay, you're going to heal it, but there is no any compensated part can be happen. If you, for example, if you cut a part of your body, it will not compensate it, and so on. So this is the meaning of capacity of regeneration. After that, we have the fourth type of asexual reproduction. Sprogony, it is the fourth type of asexual reproduction. And to learn more about sprogony or sprogony, we need to know what does it mean, spore. Well, at the first, sprogony, it is the separation of single cell. This cell is called spores, okay, and this cell that grows by mitotic division to give us a complete individual. Again, we have any individual, okay, any living organism. It will release different types of cells. One of those cells are called spores. Each spore divides mitotically to gives you a complete individual body. Well, so a spore is what? The spore, a single cell that contains a little amount of water, cytoplasm and nucleus, and don't forget that surrounded with a thick coat to protect it from hard environmental conditions. Well, and we have some examples here, like what? For example, some algae, some type of algae, or primitive, they are primitive plants. Some plants, such as ferns. What does it mean, ferns? For example, we have adiantum, many fungi, such as bread mold fungus and mushroom fungus. Again, we say we have some examples like algae, plants such as ferns. Ferns, it's a type of plant that reproduce by spores. There is no any seed formation inside this type of plants, which is called ferns. Okay, and most of fungi like bread mold fungus, a mushroom fungus, and so on. Well, let me to give you an example here. Well, the mechanism, or how does it occur, the reproduction by sprogony. Spores, I mean the single cell which is called a spore, surrounded by a amount of cytoplasm, and it contains a nucleus and surrounded by a thick coat. Spore mature, it will grows up, release from mother cell or disappear, rupture to the air. They are what? They are flying the air. After that, when they reach to a suitable medium, the coat surrounded, the thick coat surrounded, it ruptured. Ruptures mean that disappeared, it disintegrated. Okay, this coat is what? Ruptured. And after that, each spore absorbs a suitable amount of water and divides to several times by mitotic division and grew to give us a new living organism. This is the mechanism of reproduction by sprogony. And this is a production by sporogony in bread mold fungus. Well, but actually it is under microscope. Okay, the head, the large head is called sporangium. So the a large capsule that appears on the screen, this is called the sporangium, and this is sporangium. The plural form, which is sporangia. Okay, each sporangium, it contains a large number of spores. They are the sporangium ruptured, and when it ruptured, the spores are separated or scattered in the air. When they are settle on the suitable medium, they grow to give us a new individual after absorbing amount of water. We have a give reason here for the previous discussion. One, reproduction by sporogony has many advantages. Like what? Because of or due to tolerance of hard conditions, as they have thick coat. Tolerance, it means that they can tolerate a hard condition. They can adapt with the hard environmental conditions due to presence of thick coat. This thick coat, it can remain for a very long time, okay, under unsuitable conditions. And when the condition will be favorable, the coat is what rupture, and the spore cell can grow and to give us a new individual. And also wide distribution. They are light, light in weight, okay? They are very light in weight, so they can, like, they looks like as a very light or a large number of cells. And also quick propagation as a result of light in weight, so they can propagate or can distribute with a large number, so they gives us a large number of individuals. After that, we have one of the most important asexual reproduction cases, which is what? Parthenogenesis. One, parthenogenesis is a type. I know that you didn't hear this word before, the parthenogenesis, but I'm going to discuss it with you. Well, actually, it is a type of asexual reproduction in which what happens, the female gamete, the egg cell, it grows by mitotic division without any fertilization of male gamete to form a new individual. So, did you mean that the parthenogenesis is restricted to the female only, that happens by female? Yes, it happens only by female gamete, okay? Also, the parthenogenesis considered as a special type of asexual reproduction because of what? The new individuals are produced from the female gametes only, and they are similar or look like to the female gamete. So the only type, don't remember that, okay? I need you to remember that well. The only type of asexual reproduction that restricted to the female only, okay, without both types, male and female, just female, which is what? Parthenogenesis, okay? And how many type of parthenogenesis that we have? One. While the other types, male and female gametes, as we said, can reproduce, each one of them a male or female plenary, a male or female starfish, and so on. Each one of them, if you cut a part of it, it will be compensated and gives you a new individual. But in case of parthenogenesis, the female gamete only, okay? So restricted with female gamete. Don't forget it. One. For examples, some worms, some crustaceans, as we said, crustaceans like what? Crustaceans like crabs, like shrimps. They are exoskeleton, and also most of insects. Well, types of parthenogenesis. Well, we have two main types for parthenogenesis. The first type, natural parthenogenesis. And what does it mean, natural? Well, do you know that happen naturally? Okay, I mean that it doesn't happen inside the lab. The scientists, they didn't do anything, just it happens by the nature, okay? For example, the queen of honeybee, it gives many eggs, okay? Some of these eggs grow without fertilization. Seriously, yes. Some of them grows without what? Without fertilization to form what? Haploid individual. What does mean haploid? As we said, haploids mean they carry the half of genetic materials, and they are give us what? Males or drones. And but let me to connect something here with you. One, if the eggs of honey, so number one, the natural one, if the natural eggs are grows naturally, without any fertilization, they give us what? Drones and males. While if the eggs of honeybee are fertilized, if the fertilization is happening, we are talking about the sexual type, okay? What happened? It form a queens or workers according to a type of food that the honeybee had been eaten before. Get the difference? Well, this is a natural type, so we call it what? This type is what? Is sexual or asexual? Yes, this type is, yep, sexual reproduction. Okay, but on the other hand, we have another type that we can do it in lab. We can twist the living organism to reproduce asexually by parthenogens. The artificial parthenogens, which we can do it by the eggs, when the eggs of toads or some of amphibians are activated by different ways, like what? Exposure, they are subjected to heat, subjected to electric shock and to radiation. I mean, like X-ray and gamma ray, these mean of radiation. Also salt agitation, I mean a high or increasing saltness, and also by prickling with needle. If you have a needle and you have your test tube and put some of eggs of toad or frogs in the test tube, and you have a needle, okay, prickling needle, and you're going to activate the egg by it. What happened to it? The chromosomal material duplicated of the egg, and eggs grow into to complete or give us new individual. But this individual is identical to the mother cell, which is what? The female, which gives us what? Which gives us the eggs or the egg strap and so on. So we have two different types of parthenogenesis. The natural one, which happens in the honeybee, and the artificial one that we can do it. We can do it like some example of amphibians like toad and frogs. For example, frog, sea star, and for your information, the early embryonic stages in rabbit. Well, the early embryonic stages, it's mean what? Well, embryo, it's mean the after formation of fetus inside the uterus. The starting after the formation of morula, they are converted into the embryonic stage, start to form it inside the uterus of the female. This is what parthenogenesis that happen in frog, sea star, and early embryonic stages in rabbit. And after that, we have something called tissue culture. And tissue culture, from its name, so it happens in what? It happens in, yes, it happens in plants, sure. Well, it is a type of isolation of some tissues from the organism, and we put it in what? In a semi-natural nutrient medium. And also it followed by what? By a differentiation and a progress to form what? Complete individual. We have an experiment or activity to show that. Instead of saying those words, we have an activity to show that. Well, for example, we have what? It happens artificially in plants, but it is a very difficult to happen in animals. All right, well, till now it's very difficult, but maybe in the future we can do it in the future. Okay, but right now it is a very difficult. So the last type of asexual reproduction, which is what? Tissue culture. Well, let's discuss the activity that we have here. We have two examples like carrot and tobacco plants. Well, we're going to bring a small pieces of carrot or tobacco leaves. We put it in the conical flask. Okay, it's a type of flask. And this conical flask is containing what? A semi-nutrient medium, or like coconut milk. So this is the nutrients of it. It's a food. We need to give it food. So we give it directly what? Sugar or coconut milk. The nutrients elements also, it should be exist, and some hormones just to activate. From the previous lesson, you have to know that hormones, they are just what? To activate the process. So we have a conical flask containing coconut milk and nutrients elements and also hormones. Well, the tissue of the leaves of tobacco plants or of carrot, it grows to give us what? A complete plant. This is what? This is the tissue culture we bring. Just we brought a piece of plant and it gives us a final product or final individual. So it depends on what? The area of that any somatic cell. Somatic cells mean what? Cell that contains a double genetic material, or the genetic material, the full genetic material, which is 2N. It's not haploid. Somatic cell, it's mean the cell which contains the full genetic material, like cell of your body, you yourself. Any cell of your body called somatic cells, except the cells of sperms and cells of, I'm talking about the human, sperm or ovum. It just contains the half of genetic material or haploid. So it depends on the idea of that somatic cells. The organism contains what? The genetic information as the whole organism. And this is experiment. I'm going to discuss it with you. We have a carrot here, a piece of carrot. We cut it and we brought, actually it's not a conical flask, it's just a tube. And we put it a coconut milk as a kind of nutrients. And we know that going here, it shows us for you the group of cells or the tissue. And mitotically, that what happened inside it, but we cannot see it by our eyes. The group of cells, they are mitotically divided to give us what? Yes, to give us a new individual or the new carrot plant. So this is a simple meaning of tissue culture in the plant. Okay, well, we have also some application of the tissue culture, like what? We have some applications of tissue culture. Number one, propagation of rare plants. What does it mean, propagation of rare plants? Rare plants, we have some, and actually we have dozens of plants. They are absolutely extinct. They are disappeared. Okay, and if you remember that, like what, some plants here in Egypt, they are very rare, and also production of strains with desired characters. Let me discuss with you what does it mean, production of strains with desired character. For example, we have here in Egypt, we need to plant or to grow a lot of different types of wheat. Okay, so we need that wheat, but actually the wheat here, and for example, like rice, we need rice here, much more rice. But the rice of Egypt, the strains of Egypt here, the species, the type, strain here means type of the rice here in Egypt. Actually, it needs much more of water, but we can genetically, or by using the tissue culture, we can modify a different type of rice to take just a little amount of water. So we can produce a large amount of rice by using tissue culture. Okay, without doing a large effort. So propagation of rare plants, production of strains with desired characters, mean that character that we need it in our daily life. And also production of strains that resist diseases. So if you have a weak plant, it will gonna be die and disappeared, and it doesn't give you any benefit if it's weak. But we need something that, or a plant that resists and to face the most of unfavorable conditions of the environment, and also what? To resist the disease. This is the meaning of production of strains or types that resist diseases. Also, we can solve the food problem by increasing what? The production, or capacity of the plants by using tissue culture. Just you can from one plant, okay, and instead of remaining to the plant to grow up more and more and to take most of seeds from it and to grow it after that, it gonna takes a lot of time. You're going to waste your time. But just what did you know? What did you need inside it? Just you need one plant. You can cut it into 100 parts. Each part it gives you what? A new individual. So you can make forests and you can make a lot of plants and to plant a different type by what? By increasing the production by using tissue culture mechanism. And I need to know something here, that the selected tissues of the tissue culture plants, it can be stored for a very long time in liquefied nitrogen. Well, liquefied nitrogen, it is nitrogen, we can use it, and actually it's not only in the tissue culture but in the different life application. And for your information, the temperature of the liquefied nitrogen, it's about -196 degrees Celsius. Okay, it is very cold. So, but it doesn't freeze. Okay, yeah, as much as a very cold -196 degree, but it doesn't freeze. It's in form of liquid. So the liquefied nitrogen, we can store the tissue culture or any organ. Okay, by the way, inside what? Inside the liquefied nitrogen. Well, give reason for number one. Reproduction is important for species, not for individuals. Again, reproduction is important for species, not for individual. For what? Well, as we said, the reproduction is very important for species, not for the individual. To what? To prevent it from extinction, and also for propagation, and also for what? For remaining the type of the species. Okay. Think, and I need you to the next time to write the answer and to revise it. Number two, the second question. Reproduction is the least important character for living organisms. What does it mean, the least important character for living organisms? I mean that it doesn't a fundamental point in the living organism, okay? Think, and I need you also the next time to what? To squeeze your mind and to give me the answer for this question. Very easy. And also we have a question here, which is regeneration is limited in higher vertebrates. I'm going to give you the answer, but and the next time also I need you to write it. Restricted or limited in higher vertebrates, as we said, in the higher vertebrates like us, okay? It limited or restricted on what? On healing wounds and healing the cut veins or tissues, but there is no any compensation or there is no any regeneration of missed parts. But tell me in higher and lower and lower. Okay, remember that. I need you to give you the final answer in higher vertebrates and lower living organisms like hydra and starfish, and also in some crustaceans. After that, we have the last question here, and this is for you. Peer breeders, or people that interested in growing pearl, collect sea stars with great care, or they collect them and burn sea stars, not cut them. I'll give you the answer of this question. Okay, well, actually, the starfish feeds on the pearls or the animal that form pearl. Okay, so when the breeders, when the workers, I mean the people, the fishermen, the interested in breeding this type, they are collect the starfish and they burn it. If they cut it into small pieces, well, actually, and they re-throw it again in the sea, what's gonna be happened? Each part of the plant or each part of the starfish, it will give you 4 or 5 new individuals. So there's gonna be a disaster, catastrophic disaster. So they're going to burn it to absolutely remove all remainders of the starfish, because they are regenerated by regeneration. And this is a comparison for you. and I need you to just to compare between penetration and budding to give you definition and examples, and after that to tell me the mechanism in each one of them, and if there is any importance, and also write it down. Finally, we have a comparison between the penetration and budding. Okay, this is a form of the comparison that I need you to type. I mean that is it sexual, asexual, both of them asexual reproduction. Occurring rates, I mean in penetration it happens only in the unicellular living organism, while budding in both of them in unicellular and multicellular. Unicellular like what? Yes, like yeast fungus. Mother cell, I mean penetration, is it remains or disappeared as we said? Yes, in penetration the mother cell disappeared, absolutely there is no mother cell. While in budding it remains. In case of daughter cell, is it maybe separate and form individual, or it connect to form colony? Finally, uni and multicellular. Penetration, it happens in what? It happens in just unicellular, while budding, both of them, uni and multicellular. The last piece of information here, I need you to draw each one of them, just what kind of dramatic, okay? I mean, I don't need you to draw something for art, okay? But just scientific drawing.